All right, so we are on chapter 6, section 3. You can tell I'm in a different location. I'm in the lab actually right now. So at least you get some different background today. All right, so section 3 is all about stereochemistry. So all about three-dimensional arrangement of things. So if you cast your mind back to chapter 4, section 5, where we talked about chiral carbons, right? These are carbons that have 4 different things attached to them, right? So you can't have carbons that are triple bonds, you can't have carbons that are double bonds, you can't have a carbon that has 2 hydrogens, that kind of stuff. So remember that when you have something that has a chiral center, that chiral center can exist in 2 different forms, and we call those 2 different forms enantiomers. And so remember that we said enantiomers are non-superimposable mirror images. So I've drawn a molecule here, and you can pretend like there is a mirror right between the 2, right? And so there are 4 different things around that carbon. It is tetrahedral, but if I took these 2 and I superimposed one on top of the other, look what would happen. My hydroxyl group would superimpose, my CH2, CH3 group at the bottom would superimpose, but what's my problem? My problem is left and right. Left and right, I have 2 different things, so I can't directly superimpose the 2. These are non-superimposable mirror images. That's really, really, really important. Okay, so if we look at our new way to draw glucose, right? I want to know how many chiral centers are in my glucose molecule. So whenever you're counting on glucose, what I want you to do is to start counting wherever your carbonyl group is. So my carbon, my carbon with my double bond oxygen, that's going to be carbon number 1. Always, 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 that's going to be carbon number 1. So carbon number 1 has a double bond. Can't be chiral if it has a double bond, so put an X on that one, not chiral. Go to carbon number 2. Carbon 2 has all the stuff at the top, has a hydrogen, has a hydroxyl, and then all the stuff on the bottom. And so what you could say is that one is chiral. Well, if we look at all the other ones, all the other carbons are going to be chiral until we get to the very bottom to carbon 6. When we get to carbon 6 has 2 hydrogens. So this one cannot be chiral. And the reason this works, that all these 4 carbons in the middle are chiral, is because the ends are different. One, we have a carbonyl group, and one, we have a hydroxyl group. So the top's carbonyl, the bottom's hydroxyl. So now we have glucose having 4 chiral centers. So if we took one example, let's say carbon 3, right, our one kind of in the middle, what you can do is you can look at the different things. We looked at carbon 1, we said carbon 1 wasn't, we said carbon 2 is. Now let's look at carbon 3. When we look at carbon 3 has all this stuff at the top, all this stuff at the bottom, and the 2 things are not equal, so that they are different. And then left and right, we have a hydroxyl and a hydrogen. Same thing with carbon 4, same thing with carbon 5, okay? You're gonna see 4 different things attached. So the number of stereoisomers that are possible really increases every time we have a new chiral center. So if you have a molecule that only has 1 chiral center, there are 2 possible stereoisomers. Oh look, we had 1 chiral center. This is our chiral center, right? We have 2 possible. Okay, we have 2 possible stereoisomers. So anytime you're trying to figure out how many stereoisomers are present, the formula is 2 raised to the n, where n is the number of chiral centers present in the molecule. So if glucose has 4 chiral centers, right, that's 2 raised to the 4, or the same thing, 2 raised to the 4 is the same thing as 2 times 2 times 2 times 2, which is 16, okay? So glucose has 16 stereoisomers. Wow, that's a lot. But here's the cool thing. Our bodies only like 1 of those 16, only 1. And so we're gonna talk kind of specifically about that 1. So when we talk about drawing these molecules. It is really, really hard to draw them in 3 dimensions. And it's going to get really tiring if we have to draw these with all those dashed lines and the solid triangles to show that things are coming into and out of the board at us. So what you can see, right, is what we've drawn in the past. In the past, we've said, okay, if something is going away from us, it is dashed. If something is coming towards us, it's going to be solid. So if we look at our chiral carbon, this is our chiral carbon, right? This is what we've been drawing. Here I'm showing you what's called a ball and stick model. So it's really in 3 dimensions. And so what you can see with this model is, let's see if I can do this too. And I've built one as well, right? Okay, what we can see is that this is our chiral carbon. It's kind of hard to see. I think it's easier to see on the screen. See, okay, so this is our chiral carbon right here. We have 1, 2, 3, 4 different things attached to it. So I'm gonna kind of hold on to the chiral carbon, right? That's our chiral carbon. So what you can see is here, if this is the chiral carbon, coming out at you are these 2. Can you see that? They're kind of coming out at you. So these 2, see if I can do this, these 2 that are coming out at you, right, are these 2 right here, right? So that's our kind of triangular things right there, and they're coming out at you, okay? Then we have some that are going towards me, right? They're going away from you. And so that's these right here. Do you see? So I'm gonna hold on to that chiral carbon, and I'm gonna turn it. Do you see how it's kind of angled to the back? Same thing, if I hold this, same thing with this one at the bottom, right? If I kind of turn it for you and get closer to the screen, Dr. B, you can see that it goes backwards towards me. So whenever they go backwards, what we do are those little dashed lines. So that's the little dashed lines, and then here's our little dash lines, and here's our little dash lines. Gosh, that would take a long time to draw. We don't want to do that. So what we use are what are called Fisher projections. In Fisher projections, we use only straight lines. And it's really kind of cool how we use Fisher projections, because anytime we see a vertical line and a horizontal line intersecting one another, that is a chiral carbon. So not all your carbons are going to be lines, but all of the ones that make an intersection, so like this, like a plus sign, a perfect plus sign, this is a chiral carbon. So we don't have to draw those asterisks anymore. All we have to do is see that it is a solid, perfect plus sign. This carbon right up here is not chiral, because do you see how these bonds are going at an angle, right? That's a visualization for me to know that that's not a chiral carbon. This carbon down here at the bottom, not a chiral carbon, because I didn't draw it in the Fisher projection with that perfect plus sign. So we're going to use these, and it's just going to take practice. This is just one more way to kind of think about a molecule. So we said, as we said in our Fisher projection, our horizontal lines in the chiral center represent wedges. So wedges are telling us that they're going towards us. And then the vertical lines are going to represent the dashes, which are saying they're coming out at us. All right. So any chiral carbon not shown as a C, so any chiral carbon that you put, any chiral carbon, you're not going to put a C. You're just going to put an intersection like a plus sign, right? So it's going to give you a super, super easy way to know, is this a chiral carbon, yes or no? If it's a plus sign, absolutely. Now, these Fisher projections have designations. They're either designated D or L. And we're going to talk about why they're D or L. And we're going to use the model reference molecule called glyceraldehyde. Okay, so here's our 3 models. We have the wedge and dash, we have the Fisher projection, and we have the ball and stick. And so anytime that we are looking at these kinds of models, it's really easy to see in the Fisher projection. So that's these 2 here, 1, 2, right? Ooh. So what you're going to do is you're going to find your carbonyl, right? So we have our carbonyl. Now find your chiral carbon that is farthest away from that carbonyl. Well, I only have one. There's only one chiral carbon, and that's this one right here. So if we designate it as L, it's going to be super easy. The alcohol is going to be on the left. So L, alcohol on the left, right? Okay, D, look at D. I'm going to find my carbonyl, and then I'm going to find my farthest away chiral carbon, and I'm going to put my alcohol on the right side, okay? That's the only difference between left and right. All right, we're going to practice it. Don't worry, we're going to practice it. Okay, and we already went over that. All right, so D sugars, this is what we just said, have the OH on the chiral carbon farthest away from our carbonyl on the right side. So D is on the right and L is on the left. And so we're gonna use this and we're gonna look so that we don't have to do all these wedges and these dashes, right? We're going to do our Fisher projection. So here's our Fisher projection. Isn't it so much easier to look at? I look at all these individual plus signs and say, that's a chiral carbon, that's a chiral carbon, that's a chiral carbon. So I have 4 chiral carbons in glucose. It was super easy for me to pick that out, right? And so how do I know what orientation this is in? I found my, my carbonyl, and I'm gonna go to my, whoops, and I just erased. I'm going to go to my chiral carbon that is farthest away, and I'm going to say, is it on the left or is it on the right? And this one is on the right, and so therefore it must be the D orientation. Okay, our bodies like that D orientation. Our bodies use that naturally as sugar. So that's what our bodies are really, really, really used to. Okay, now we talked about enantiomers. Enantiomers are those mirror images, right? Non-superimposable mirror images. So it's very easy to draw a Fisher projection that is a mirror image. So let me show you. Anything that is on the original molecule that's on the left, we're gonna put it on the right. Anything that was on the right, we're gonna put it on the left. No big deal, okay? Ooh, my palm keeps wanting to slide these things. Okay, so I'm gonna say everything here, this is on the left. Here, well, let me do a different color. Here is going to be on the right. So we don't mess with anything at the top, anything that's not a chiral carbon, we leave alone, right? So don't mess with this stuff and don't mess with that stuff. So I'm going to uncircle it because we're not going to mess with it. You would just basically copy and paste that. Now, everything that was on the left now is going to be on the right. And I just drew to the left again, to the right. Okay, there's my stuff to the right, right? I have hydrogen, hydroxyl, hydrogen, hydrogen. Now, everything that was on the right is gonna go on the left. So I have hydroxyl, hydroxyl, hydrogen, hydrogen, hydroxyl, 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 hydroxyl. So if you put these two right next to each other, it's really easy. This is what I do. I start on this side. So all I have to do is copy over, and it's pretty easy. And so we have D glucose versus L glucose. And how do I know which one's D, which one's L? I know that if I look at my carbonyl and I go to my chiral carbon farthest away, I see that my OH group here is on the right, and that makes it D. Okay. All right, so drawing it, locate your chiral centers and switch your horizontal groups on your chiral centers. So let's try that. So I'm gonna cheat a little bit. I know that these molecules are gonna be exactly the same except for what's on the sides of our chiral carbons. So what I'm gonna do is I'm going to just copy and paste it. Now I'm gonna find my chiral carbons. My chiral carbons are 2, 3, and 4. So on 2, 3, and 4, I'm just going to erase my stuff that's attached to my chiral carbon. Pretty nice, right? Okay, now all I have to do is take the stuff that's here and put it right there. So instead of writing OH, I don't want to do that because now it looks like I'm bonding to the hydrogen. So I don't want to do that. I got to hide my hydrogen again. Okay, I want to write oxygen bound to a hydrogen. We still call it hydroxyl. We're just showing you that the carbon's attached to the oxygen. Okay, OH, and then I'm going to put my hydrogen, and my hydrogen. Okay, so this is an enantiomer of ribose. And I know that this is D because my hydroxyl is on the right, the farthest one away, right? Here, this one, farthest away from my carbonyl, it's on the left, so this must be L, ribose. Okay. All right, so you can have stereoisomers that are not enantiomers, right? We said there were 16 different possibilities. How do you get that many different possibilities? Well, the other possibilities are not exact mirror images, and we call them diastereomers. So anytime you flip 1 or 2 or 3 of the different chiral carbons, but not all of them, you're going to be a diastereomer. So it's not an exact mirror image. So let me show you an example. We have D-glucose, right? So if I compare D-glucose to D-galactose, right? This stuff is exactly the same. So we don't worry about that, right? Okay, now, first here, hydrogen and hydroxyl, those are the same. Hydroxyl and hydrogen, the same. Whoa, look at this. Here's a swap, right? So those are swapped, and then hydrogen, hydroxyl, hydrogen, hydroxyl. So D-galactose is a diastereomer of D-glucose. So this one is a diastereomer, D-E-R-E-O, diastereomer, okay? Let's try that with the other one. I'm gonna kind of erase because I got a lot going on here, right? Okay, now I'm gonna check this one versus this one. Are they the same? No, they're not the same. Okay, how about this one and this one? Oh, they're the same. How about this one and this one? Oh, they're not the same. And how about this one and this one? They're the same. Okay, so I have 2 groups that are off. This is another diastereomer of D-glucose. Okay. All right, so when we look at these, I've only showed you a couple of different monosaccharides. This is kind of most of the, not most of them, but the ones that are 3 to 5 carbons. So these are D monosaccharides that have 3 to 5 carbons. So we start out, oh, and I'm in a room that has motion sensors and they're not picking up me moving enough. All right, so our first one, D-glyceraldehyde, this is our easiest one because it only has 1 chiral carbon. What we do to go from glyceraldehyde to our next one is we add another chiral carbon. So here we've added another chiral carbon. The only difference between the two, right, is we flip that hydrogen and that hydroxyl. So these are D-erythrose and D-threose. Those are diastereomers of one another, okay? What do we do when we go to the next one? We can add another chiral carbon. Okay, so D-ribose and D-arabinose are, guess what, diastereomers of each other, right? And so on and so on and so on and so on. There are a couple that are really important that we're going to talk about in detail, like D-glucose, like D-mannose, D-galactose, and we're going to talk about a few others. But there's one thing about this I do want to mention. When you're looking at these, I don't particularly care for the way they do this because they put this group up at the top, CHO. It's important that you remember that these are a carbonyl carbon. So that CHO is really carbon double bond to an oxygen with a hydrogen, and then the rest of the molecule, this is the R, right? It's a terrible R. But it's not single bonds, even though it kind of looks like it, but it's not. So just be aware of that when you're looking at these structures. Okay, so let's talk about some important monosaccharides. Glucose. This is the #1 most abundant found in nature. Sometimes you'll hear it called dextrose, and sometimes you'll hear it called blood sugar. So when you're in the medical field, you hear it called blood sugar. We find it in fruits, in vegetables, in corn syrup. And so our bodies use this glucose, we break it down inside of our cells, and we produce energy. And that process of breaking down glucose to make energy is called glycolysis. So you've probably heard of diabetics, right? Diabetics have a really difficult time getting glucose from the bloodstream into their cells so that glycolysis can occur. They can do glycolysis, but what they have trouble with is getting glucose into the cells. And so they have to be really, really careful and monitor their blood glucose levels or their blood sugar levels, right? Another example of glucose is inside of sucrose, our table sugar. So like if you're going to bake something, you're going to put sugar in your coffee, whatever it is, you're putting table sugar. Table sugar is two monomers that if you cleave it, one of the ones that you're going to get is glucose. It's also one unit inside of lactose, which is the sugar that's found in milk. In amylose, amylopectin, glycogen, cellulose, it's in a bunch of them. So it's very, very important. The next important monosaccharide is called galactose. Galactose often likes to combine with glucose, and this is our lactose. This is our disaccharide lactose that we find in milk. Galactose also has a single chiral carbon, and it's arranged oppositely that of glucose. And so you can go back and look at all those structures, right, and compare them. So when we're talking about different molecules that differ in just one chiral center, like galactose and glucose, we call those epimers. And so the body can actually take an epimer, so galactose is an epimer, and convert it into glucose with an enzyme, and then we can survive off of galactose, right? But you need that extra enzyme. Okay, mannose. Mannose is a monosaccharide. You find it in cranberries. That's where it's most easily found, and it's really difficult to be absorbed by the body. And so it's been kind of the common treatment, over-the-counter treatment that doesn't really work to treat urinary tract infections due to the excess mannose that's inside of the cranberry juice that's going to flush through the urinary tract. But recent studies really now question whether this is really effective and good. So more to follow. Fructose is another really important monosaccharide. This is a ketose fructose. So what's a ketone? A ketone, right? Key, you need two, right? You have R and an R and our double bound. You need the lock in the key, right? That's two of them. So what we have here is one R group, and then here's our other R group, right? So be careful that you remember what that means. So this is our fruit sugar, fructose, fruit sugar. That's where it comes from. And sometimes it's called levulose. It's found in fruits and vegetables and in honey. And so usually what we do is we combine this with glucose, and that will form the disaccharide sucrose. So fructose is really the sweetest monosaccharide. It's 1.5 times sweeter than our normal table sugar. So our perception of sweetness is different depending on the conformation and the structure of the sugar. So it's not an epimer of glucose, right? Because this one is a ketose. It can't be an epimer, but it can be broken down for energy by the body. So this is another one that our body can use. So do you kind of need to keep track of the ones that our body can use? Absolutely, absolutely. All right, ribose. Ribose is really important. Ribose is a 5-carbon sugar, so it's really a pentose. Ribose is just like another name that we use, and you'll hear it. You've heard this before. I know you've heard this before when DNA and RNA. So deoxyribose is a part of a nucleic acid. That's why we call them RNA, ribonucleic acid, and then deoxyribonucleic acid. So they're very similar, except you could hear there's ribo and deoxyribo. So when we're in DNA, the only thing that's different is that on carbon 2, here's carbon 2, and here's carbon 2. On ribose, right, we have that alcohol. On deoxyribose, we have another hydrogen. So they're both sugars, they're both pentoses. What's different is that carbon 2 does not have a hydroxyl group on the sugar, which is why we call it deoxyribonucleic acid, DNA. All right, ribose, oh yeah, is also found in a vitamin, in riboflavin, and some other not quite so important biological molecules. All right, so one of the things that we have to do is we have to be able to tell the difference between an enantiomer and a diastereomer. So when you get to questions like this, you should kind of jot down to yourself, what is an enantiomer, what is a diastereomer, before you even look at the question. So I know an enantiomer is a stereoisomer that is a perfect mirror image, right? Where everything is just flip-flopped. A stereoisomer is at one or more of our chiral centers, we have a flip-flop, but not all of them, okay? So let's see what we get. We're gonna compare mannose to structure of A. So if we look, right, we're not worried about the top stuff because it's exactly the same. Now, if it was not exactly the same, they're neither enantiomers nor diastereomers because they're completely different molecules. But we're already assuming that they are. Okay, so we have comparing this one and this one, they're the same. This one and this one, they're the same. Whoa, here we go. Let's try a different color. Make it a little easier to see. All right, this one is different. Here we go, right? They've been flip-flopped. And then these are the same. Okay, so what do we call these? Do we call these enantiomers or diastereomers? We call these diastereomers. Okay, now let's do the same thing, except I'm gonna erase the stuff off of here first, and I'm gonna check. Oh, when I look at my first one to my first one, they've been flip-flopped. All right, the second to the second, they've been flip-flopped. Third chiral carbon, been flip-flopped. Fourth chiral carbon, they've been flip-flopped. So look at this. I circled all of my chiral carbons. That must mean that these are enantiomers. Because if I look at everything here, HHOH, and I look here, HHOH, so everything's been flipped. That's a mirror image, that's an enantiomer. Okay, my last one here, I'm looking, I guess I should erase this one, okay. I'm looking here and I have OHH, so that's the same. Then I have OHH here, and then I have HOH here. Oh, so that's been flip-flopped. This one's been flip-flopped, and this one's the same. So this is another diastereomer because it's not a perfect mirror image, but one or more of the chiral centers has been flipped. Okay, and then our last one, I guess I should have put this earlier, but it's okay. Glucose and diabetes. So glucose is super, super important. And if we can't regulate the amount of glucose in our bloodstream, then we end up with a disorder called diabetes. And so the way that our bodies regulate the amount of glucose inside of our blood versus inside of our cells is through hormones. There are two hormones that are super important. The first one is insulin. So let's say you eat a meal. That means that you have consumed some amount of sugar, right? And it's gone into your bloodstream. So your body's gonna produce insulin, and the insulin is gonna tell your body to pick up the glucose. So if you pick up the glucose, that will bring your blood sugar down. So what happens is, right, if this is your blood sugar, right? You eat a meal, it's gonna go up. When it gets up here, your body's gonna say, make insulin, right? Your body makes insulin. Well, where does that blood sugar go? That blood sugar tells the cells in your body, you need to take up the glucose. And so then your blood sugar is gonna drop until you get hungry again. And then you're gonna, oh, I'm really hungry, I need a snack, right? Or it's time for lunch. And then your blood sugar is gonna go up. And so your body does this all the time. It's an up and down, up and down. So anytime you're peaking up here, you're making insulin. All right, so now what happens when you go to sleep? You sleep for quite a long period of time, right? And so you're going for a prolonged period of time with low blood sugar. So what are you gonna do? Your body's gonna make a second hormone, and that second hormone is glucagon. So when your body makes glucagon, it tells your body, oh, I know you have some glucose stored inside of your liver. You can go ahead and break that down and put it into the bloodstream so that I can have a moderate level of glucose in the blood. And so when your blood sugar gets down low, your body is making glucagon. So these are the points where your body is making glucagon, okay? So if you are diabetic, you can't do this regulation as well as everybody else. So about 10% of the US population, that's a big percent, has diabetes. And so the way that we designate whether someone has diabetes or not is if they have prolonged periods of high blood sugar. So if they have prolonged periods of high blood sugar, we consider them diabetic. There are a couple of different causes, right? We know that the sugar's in the blood and it's not going into the cells. So there's some kind of disconnect there. The cells aren't bringing the glucose from the bloodstream inside of them. So there are two ways that that could happen. Number one, your body does not make insulin. That's type 1 diabetes. So either you don't produce any or you produce some, but it's all about insulin production, right? So if this is your problem, you have to take external insulin. So an insulin pump, an insulin injection, something like that. You have to get your hormone from somewhere else because your body's not making it. In type 2 diabetes, your body makes insulin just fine, but your cells don't recognize that insulin as a signal. And so even though you've eaten, your body's produced the insulin, your cells still aren't taking that sugar in. And this is really about 95% of the cases of diabetes, and that's type 2. And so a lot of times, type 2 diabetes can be controlled with diet, exercise, weight loss, and medication. You don't have to have things like daily insulin injections or things like that. Okay, we are gonna go and do some practice problems. So let me pause this. Okay, here are our practice problems. So first, we're identifying monosaccharides as either the D or the L isomer. So remember, in order to identify that, right, we are going to, let's see. So if we want to know D or L. So remember what I said, kind of write your instructions. What are you supposed to be looking for before you go and look at all these crazy molecules, right? So you want to look for the hydroxyl on the farthest chiral carbon from the carbonyl group, right? If it's on the left, let's see, if OH is on left, that is L. If OH is on the right, we call that D, okay? So that's your little ticket. You can write that on your scratch sheet on the exam, you know, make it work for you. Okay, so here's my carbonyl. I'm gonna go to my farthest chiral carbon, and I remember my chiral carbons are only the ones where I have that perfect plus sign. So I only have 2 chiral carbons in this molecule. So I go to my farthest one and I look at my farthest one. Is my hydroxyl group on the left or on the right? It is on the right. So this is a D isomer. Okay, let's do the same thing. Here's our carbonyl. I want to go farthest carbon. 1, 2, 3. That's our farthest chiral carbon. Here we go. Left or right? Left this time. So this is the L isomer. L isomer. Okay, here we go. Here's our carbonyl. We're going to go to the farthest. Where do we see it? We see it on the right. So that is a D isomer. Once you have the instructions and you know what you're doing, it's not so bad. But if you miss those instructions, if you can't figure out what to do, that's where you're going to have the most amount of problems. Okay, so you might want to make yourself some flashcards like, how do I identify a D or an L isomer? And then put your answer that I just wrote here in blue as the flip side of the card. Something like that. All right, let's see. And I've somehow managed to get this on 2 pages, but it's okay. Draw the Fisher projection for the enantiomer mirror image. Sorry, y'all. Hang on.